Hello, my name is Olivia Dlugi, and here I'm going to be presenting my Fly Lab final project. So let's begin. So to introduce the unknown mutants, here we have a figure of the wild type beside the Bib and Sun, which are both of our two unknown mutants. And here we see Bib has kind of like a dull red with gold flakes, and Sun more of a bright orange, and then comparing that to the wild type. So when doing our crosses, um, to kind of gather information, whether it's X-linked or autosomal, we had to perform these genetic crosses between the mutant wild type Drosophilas, setting up the P1 crosses using virgin females, and then counting the phenotypes of the offspring separately for females and males. So we needed to use virgin females because it's crucial that they are not inseminated, because as I said here, this can store sperm from an earlier mating, which can be devastating to the results because we don't know what male that was. It could have been a father from an unknown male, and previous vials, it wouldn't give the data we need. And then sex specific crosses, like identifying whether they're male or female, incredibly important so we could see if it's X-linked or autosomal. So we're trying to navigate through this by looking at inheritance patterns, uh, through comparing the phenotypic patterns, like I said, deriving whether it's X-linked or autosomal. So I did this through clearing the vials after a week, usually, and uh, every 6 hours looking if there were any females that regenerated to again avoid the females being inseminated. And then I would lightly etherize them, isolate them, the females and males, and do this through identifying sex chromes, which are on the male legs, which is usually like a black dot, and just continuously gather more and more until at the end we put in a male to get the cross. So here's our data I got at the end. We chose our bib cross because we were having issues with the sun vial. It just was not, um, they weren't mating. Our flies were just sitting there. I was going continuously week after week. Well, only like a couple weeks because then it would be too late. But yeah, they were in mating. So just to make a conclusion, which it could not be, is not very accurate. I just said it's X-linked because we had a bunch of females in there. Um, but of course, we weren't able to get offspring. So then here we have 18 and 21. So we see that it's an equal, I guess, amount. So it's autosomal. We were able to conclude it's autosomal. So 3-point mapping, our unknown is the bib. And how would you have performed the 3-point mapping if we had time? So we would have crossed the parents so that the F1 females are heterozygous at all three loci, and then cross those F1 females to the tester males, which would be this genotype. Um, then map frequencies like by hand, NCO when we're gathering the data, and SCO and DCO, just like we've been doing in class. And here's our simulated data. Again, we're able to get the NCO, DCO, SCOs, and find the middle gene, whatnot. Then to be able to derive this, we got our genetic map, um, the L, the lobe eyes, 30 map units from our unknown mutant, which we're trying to find, and then the unknown mutant 3.5 to pin. So this will be a total of 33.5 length. So from that gene order, we were able to compare to the chromosome map we were provided. And then with the information of our pin, how many map units that is, able to kind of include that here where I'm showing with the cursor right here. I know I wrote a lot here, but I'm just gonna go through how I was doing this. On the reference map, I kind of just subtracted 106, which was the pin map units, from the 72 lobis and got 34 map units, which is extremely close to what we got here. And then from that, just add 72 to 30 because uh, the lobis would be 72 and the map units here we got to the unknown was 30, which it comes out to be 102. So since it's 102 map units, this sits between 102 and 103. I kind of went here and found brown eyes to be the closest. Um, yeah. And then we could also understand that this is on the same chromosome since they're linked due to the recombination frequencies we were able to derive. And then relating back to figure one, further supporting that, oh, brown eyes. Sorry, I didn't say that it was brown eyes. I was just kind of pointing at it. Brown eyes is the conclusion um, of the unknown mutation because of what I was mentioning before. And relating back to figure one, it was described as a dull orange. We can kind of see how that's like brown too here. Like it feels like it's very brown. So if that further supports the hypothesis. So moving on to complementation, sort of just how we were doing practice problems in class, I kind of just thought of it that way, how we can relate that to an experimental design in a way. So I would just cross unknown mutant stock to homozygous ones to the gene responsible for phenotype on the same chromosome. So for example, homozygous brown crossing the unknown to determine if BIB is on the same gene. And then if the two mutations are on the same gene, there's no complementation, and F1 offspring show mutant phenotype. On different genes, the offspring would show wild type because it carries one working copy of the other gene. Okay, so just to reiterate what I was saying before, um, I was able to determine the linear order of the three loci through three-point mapping from the simulated data and the recombination frequencies, which all supported in me finding that it's the brown gene. So we were able to find that it's which is the U relative to the chromosome map by looking at the distances, comparing um, future directions. What else do you want to know about your gene? So I specifically wanted to see the change in DNA. What change in DNA does the allele carry in the brown gene? And what effects can this have on protein function as a pigment transporter? So through looking into like the brown gene, which is it encodes a member of traffic ATPase. So it's a transporter and it's necessary to import guanine and other pigments to the eyes. So I was just wondering how effects in the function can affect it working as this transporter. So we were talking about Sanger sequencing, I believe, in lecture once, or through one of the many methods that are able to be used. And I was wondering if that could be somehow used. So we'd first have to collect flies through raising wild type and mutants, design primers to amplify coding exons, using this brown sequence, and then compare sequences looking for missense, nonsense, um, frame shifts. And then from there, if it's like a nonsense mutation early on, we can kind of infer this would be a strong loss of function. And I found this to be close to the GD EWAR experiment, um, which used nucleotide sequencing of white and brown alleles to link specific amino acid changes in the transporter. So by um, showing the sequence level characterization can reveal structure function relationships in these transporters. So applying this strategy to our unknown allele, we could kind of determine what kind of molecular, I guess, lesion it carries and how that might change the protein function. So contributions, I wrote a lot here, but basically I just did the tending of the flies and she did the assignments in class, just being aware of time. Yeah, I just did, I went in every 6 hours, did most of the husbandry and provided pictures. Everything with the flies I did, essentially. So yeah, these are my references. So I guess a major takeaway from this lab, it was cool to see how like ideas like linkage and mapping can come from direct hands-on work with living organisms, um, and can strongly affect like timing and like errors and data can really affect data and conclusions. And it was cool to see how setting up the crosses ourselves, like getting the phenotypes and everything, seeing firsthand how maybe genes don't assort independently and maybe travel together. Yeah. So thank you for listening.